Right, hi everybody, good afternoon. I'll just use this opportunity to do another quick video, in this case on the subject of indicator species. An indicator species is a species where if it's present in your water that you're investigating or your water body, um, some species are indicative of that water being very clean. Some species, if they're present, are indicative of that water being polluted. On a very simple level, you can see above my head, in a temperate river, if you sample it and you find a lot of stonefly larvae or stonefly nymphs, you can be fairly confident that water is clean, non-polluted, because stoneflies are very pollution sensitive and therefore they will die if the water is polluted. On the other hand, if your river or your water body is dominated by something like tubifex worms, bloodworms, they are very pollution tolerant, so they'll take over an ecosystem if that water is polluted. So stonefly would be an indicator of clean water. Tubifex worms would be an indicator of polluted water. Very, very simple. Up to now, we've only looked at abiotic ways of measuring pollution in water bodies. So things like, well, you could measure BOD. That would be a measurement of how much organic pollution is in a water body. You could look at something like the pH. You could look at concentrations of certain chemicals, like we mentioned phosphates and nitrates. Now, those are all abiotic factors. They're all measurements of non-living things. This is the first time where we're looking at a measurement of living things. In some ways, that's advantageous. In some ways, it makes it a little bit more difficult to measure the pollution directly. At the end of this video, I'll look at the advantages and disadvantages of using indicator species. And specifically, I want to look at something called a biotic index, where you collect data on the species that are present and you can give your water body, your river maybe, a score between 0 and 10 based on whether it's very polluted or very unpolluted. 10 being very unpolluted. Okay, let's take a quick look at what the syllabus says about the use of indicator species. I'll read through it very quickly. Under knowledge and understanding, the IB says that, well, some species can be indicative of polluted waters and therefore can be used as indicator species. That's pretty much what I've already said. And then it says here, a biotic index indirectly measures pollution by assaying or investigating the impact on species within the community according to tolerance, diversity, and relative abundance. So therefore, when you're doing an index, a biotic index, you're not just looking at which species are there and which species are not. You're looking at the biodiversity of your within your ecosystem that you're looking at, and you're testing against how tolerant those species are of pollution, how diverse they are, and how many there are in individual numbers as well. Okay, and then under applications of skills, it very simply says you need to evaluate the uses of indicator species and biotic indices in measuring aquatic pollution. At the end of this video, I'll just look very briefly at advantages of using this method of sampling a river or sampling a water body versus the disadvantages. And then in the class, we'll use that to come up with an evaluation. Okay, so indicator species, well, let me read through this. It's pretty much what I've already said. Certain species, we usually use invertebrates, as you can see here, because the small animals that are relatively immobile where they live, and therefore you can use them as pretty good indicators. High biodiversity of species within an ecosystem are usually evidence that there's an absence of organic pollution, or inorganic pollution actually, and low biodiversity is the opposite. If the biodiversity is low, that means the only species present in your water are species that are very tolerant of the pollution that's causing the biodiversity to decrease. There's usually a direct link between BOD concentration and biodiversity of indicator species. You can link one to the other. High levels of organic pollution lead to a low number in the biotic index. And then I've said here, although I will evaluate this, it's a quick and direct method of evaluating levels of pollution. Rather than collecting samples, sending them to a lab, in the case of your BOD test, waiting maybe 5 days for your results, you can see the effect directly on the species that are living at that site. So therefore, it is quite a quick and direct way of seeing if there's been an impact of any kind of pollution on an ecosystem. Okay, so I want to go through how you actually carry out or how you measure pollution on a biotic index. The one I've chosen is the Trent Biotic Index. It's named after the River Trent in England, which interestingly, again, is relevant to me because it was the first river that I worked on. My first job was with the Severn Trent Water Company, and I was actually looking at the Trent River pollution within it. An index, as you probably already know, is a number with no units on a scale where you can rank whether something is at one end of the scale or the other end of the scale. A Trent index of 1 or close to 0 would be very polluted, and a Trent index close to 10 would be very unpolluted. Incidentally, the Trent index, obviously it comes from England. I think it was one of the first indices. It was invented in the 1960s. And obviously it's relevant to rivers in England in a temperate climate. It's no use going to tropical rivers and using the same index because those species don't live there. So different types of indices are relevant to different types of rivers, different types of biomes. Okay, so bearing in mind this is relevant to a temperate climate like England. These are the species listed down here that we're going to look at. By the way, this is I've shortened the index here. The index itself has, I think, 17 groups, 17 species or 17 groups within it. So I've cut this down to 5 or 6. So just be aware there would actually be more groups here. At the top, as I've already mentioned, you'd have this guy, which is a stonefly larvae, which is, if it's present, is indicative of very clean water. And on the bottom end, you've got these tubifex worms, which are indicative of polluted water. In between, you've got things like gammarus, freshwater shrimp. This is a mayfly larva, so it's going down in the same order. This is a case caddisfly, so it's a larvae that builds itself a little tube to live in, and again, it's indicative of fairly clean water. These are other crustaceans. This is a celius, and so on. So let me go through what you do. So first of all, you identify a piece of the river that you want to sample. Usually it would be a river. And you find a riffle. So a riffle is a shallow part of the river where there's stones and things where these things might live. You put on your welly boots, you wade out into the water, and you take a net and you do something called kick sampling. And you have to try to be as consistent with this sampling method as you can. So you take your net, you put it facing upstream, and you downstream. You put it downstream, and you kick into the net. I think something like 10 times is the normal method. 10 times. You kick sediment into the net. I think you wait 3 minutes normally, and then you scoop up your net, take it to the banks of the river, and you empty out your whatever you've collected into petri dishes. And then you start to identify which species you've found. So again, bearing in mind this is only a short version of the list, what you do is, first of all, you would identify how many of these different groups in this left column you've identified. So stonefly nymphs, it represents a bit of one group. This is one genus. You might find more than one species within that group. Mayflies, caddisflies, and so on. You identify how many groups you found, and then you go to the next column. So, for example, if you found stonefly nymphs, and you've got more than one species, you already are in the top row. Yeah. You identify your top species, and you identify how many species, or how many groups, I should say, in total you've identified. So, for example, if you identified 11 of these groups present, that would be very, very diverse, so you'd expect a high biotic index. And your highest species on the list is, stoneflies. That river that you've just sampled, you would give it a biotic index of 9, which is very, very clean. So that's the way it works. That's how you use a biotic index. So just therefore to summarize what I would say are possible advantages and disadvantages of measuring pollution in this way. Well, first of all, I've already said it's a fairly quick method. It's direct, so therefore if there's an issue with pollution in your river, you can see a direct effect on the species themselves. No need to take samples of water to a lab or wait 3 to 5 days for your results to return. It's fairly straightforward to sample. The effect of pollutants can become very, very obvious very, very quickly, so therefore that would hopefully give you time to take steps in terms of pollution management. And you're actually looking at the effects on living things directly. It's not an indirect measurement, so there's no kind of interpretation necessary. You can see the effect straight away. It's a fairly easily understandable method of measuring pollution. In terms of disadvantages, well, one, it would require a fair level of expertise. These things are fairly small, not that easy to identify. So you do need some training in actually identifying these things. If you measure one part of a river in this method, using this method of indicator species, it doesn't necessarily give you a whole understanding of what's happening in the rest of the river. You have to bear in mind that it's only a snapshot. Although you could say that's a disadvantage of abiotic measurements of pollution as well. What was I going to say? Okay, one other thing is I would say there are other reasons apart from anthropogenic pollution where these things might decrease in biodiversity. So, for example, if you sample at a season where the stoneflies are not present, if they've already started to metamorphosize, then you might get a false positive for pollution. It might seem that the pollution is decreasing biodiversity when there's other reasons, biological reasons why that might happen. Actually, I've already mentioned this as well previously. It's a good idea to do these kind of tests in combination with abiotic sampling. So I did mention there's a fairly good link between the biotic index of a river and its BOD. So therefore, it makes sense to do abiotic sampling and biotic sampling at the same time. Okay, so I think that's about it. We'll discuss this in terms of evaluating it as a method in the classroom. But in the meantime, I hope that made sense. I'll speak to you very soon. Okay, take care, everybody.